Fred, perhaps you're on mute. Oh, he's not in the room. I'll come back. Elena Flowers.
Present.
Judy Gasson. David Higgins.
Present.
Vito Imbasciani.
Present.
Pat Levitt. Shlomo Melmed.
Present.
Carolyn Meltzer.
Present.
Chris Miaskowski.
Present.
Lauren Miller-Rogan. Marv Southard. Carol Watson.
Present.
And let me just look. Fred Fisher.
Yes, I'm here.
Great. Thank you. Great. Thanks very much, Fred. Or excuse me, Mark.
Okay. Thanks, Scott. And then with that, I'd like to turn the meeting over to JT.
Thanks, Mark. Good morning, everybody. As you know, we've been in the process since late last year, actually, of determining how best to deploy the remaining $3.8 billion in CIRM funding authorized by Prop 14. And towards that effort, we've been engaged in a very extensive process, which we call the strategic allocation framework, to derive a set of goals and recommendations for the board to consider ultimately at our September board meeting. That process has involved a great many meetings along the way with various members of the Science Subcommittee and the Neuro Task Force, and has taken all of that input and incorporated it into the evolving product that we're putting together for ultimate consideration in September. Rosa has most ably led this effort on behalf of the team, and of which virtually every member of CIRM has had a part in putting things together to get us to this point, and which will be involved in getting us through the finish line in September. So today's meeting is the latest in the series with here the Joint Neuro Task Force and Science Subcommittee. We're greatly looking forward to your input, and I will turn things now over to Rosa to give the presentation on this latest set of goals and recommendations. Rosa.
Thank you, JT, and I want to thank the co-chairs of the Neuro Task Force, Dr. Carolyn Meltzer and Dr. Pat Levitt, as well as the chair of the Science Subcommittee, Dr. Mark Fisher-Covry, for their support and help in developing these goals. And as I go towards sharing, let me know, can you see the presentation?
Yes.
Yes, we can.
Great. So I also want to especially thank Drs. Abla Krisi and Dr. Shyam Patel, who have been integrally involved in the development of these two goals as members of the leadership team. So without less preamble, on behalf of CIRM, and that means a lot of members of our team, I'll be presenting today's presentation. And to ensure ample time for discussion, the background and the strategic allocation framework overview will not be presented during today's meeting. As a reference, these sections were previously presented at the June 27th and July 11th meeting. And you have a couple of links in the presentation, so you can go to the YouTube CIRM video to get into those links directly at the points of this presentation. So during today's presentation, we will go over a quick review of goals 1 and 2 that were presented back in July 11th. And these correspond to the category of accelerating discovery and translation pipeline. And we will present the feedback that was incorporated from the July meeting, Science Subcommittee and Neuro Task Force joint meeting. We will then proceed to go over goals 3 and 4, corresponding to category of cell and gene therapy approvals, and that will be followed by a discussion on next steps. And feel free to interrupt me if you have any questions. So this is just the slide to say where are we, and you can always look at it on the top right part of the slides. It is a guidance of where we are. So now we are on goals 1 and 2. So as a reminder, here are the 2 first goals pertaining to the first category, and the joint meeting of the Science Subcommittee and Neuro Task Force providing recommendations. We provided the recommendations for those 2 goals. And those recommendations were met with a strong approval, not formal approval, but there was agreement with those goals. And the board members not only appreciated the thoroughness of our initial proposals, but also provided very insightful feedback that has been since incorporated into our updated recommendations. So with regards to this first goal, there were 2 recommendations. The first recommendation was to support comprehensive discovery research through structured initiatives like the DIS4 and DIS5 pilot programs that we've just started this year. We've had the first of them focused on neuropsychiatric diseases, and that's the DIS4, aka Remind, when we talk about neuropsychiatric. And the approach is to encourage collaborative, multidisciplinary innovation in stem cell and genetic research across diverse disciplines and disease indications. And the board underscored the importance of ensuring that the research findings are reproducible, particularly when transitioning discoveries from academia to industry. And they recommended that we engage industry early in the research process to help address issues of reproducibility and scalability. This is critical for the successful translation of this finding into applications. The second recommendation had to do with data science collaborative efforts. We recommended to establish a data coordinating and management center to streamline the management of data to enhance the utility of cross-disease data. And the approach could be to fund and develop a central hub for data coordination, facilitating better integration with consortia and research initiatives. And the board supported the idea of establishing this DCMC and the initiative could also enhance the reproducibility and validation of some of the results funded through CIRM, which is pivotal to move research forward. But the board also suggested that we explore the potential for dedicated data science grants to enable these collaborative efforts. And this could be aimed to leveraging the vast data sets managed by the proposed DCMC to maximize the utility of what we are funding. So this feedback will be fully incorporated and delineated into the specific granularity of the concepts if the board approves these recommendations at the September board meeting. The second goal under the first category, which is discovery and translation of therapies, is accelerate the development and utilization of X technologies that demonstrate improvements in safety, efficacy, and quality of cell and gene therapies. And the CIRM team recommended to pilot the CIRM team recommended to pilot an infrastructure technology program to bridge the gap between research and commercialization by fostering partnerships between academic researchers and industry professionals. And the approach could be to support multi-stakeholder technology incubation programs to achieve defined technology readiness levels, which could facilitate then the rapid application of cell and gene therapies. And the feedback received regarding this infrastructure technology platform emphasized the importance of fostering industry academic collaborations as well. So again, it's the same topic, but I think we are seeing a very common theme. And in this case could be to refine, scale, and commercialize the new technologies. And the board feedback highlighted that different types of industry players, including small technology innovators and large biopharma companies, could play complementary and crucial roles in this effort. So we will be also incorporating this into the concept for your consideration if this goal, obviously, recommendation is approved in September. So before I go into any questions, because this pertains to goal 1 and 2, would you like to stop for questions here, or we just go to the end, Scott? Oh, sorry, I should ask Mark. Sorry.
I think it would be helpful to continue with the discussion unless there's other thoughts.
Sounds good. So I'll keep going. If anybody raises the hand, I can stop. Thank you, Mark. So as we continue to drive innovation within regenerative medicine, one of the major goals that CIRM faces is the wide spectrum of diseases from rare to common. And this is in the context of the cell and gene therapy approvals, the goals for cell and gene therapy approvals. And for this, we have developed 2 goals. Historically, our efforts have predominantly targeted rare diseases, which has allowed us to make significant strides in areas that often lack attention and funding. And by concentrating in these conditions, CIRM has actually catalyzed advancements in the translation of these findings into clinical applications at a time when it was the time to do it. The field was ready to do this in rare diseases. And we are at a point where the largest proportion of the projects that will be ready to BLA in the next 2 to 4 years correspond to therapies targeting rare and ultra-rare diseases in our portfolio, in CIRM's portfolio. So in this slide, we outlined our preliminary goals 3 and 4, which are geared towards not just maintaining, but accelerating this momentum and finding opportunities for our population in California. The 2 goals that we have developed focus on, first, advanced rare disease projects to BLA by leveraging gene editing technologies, scaling to rare diseases, for example, and regulatory innovation. This goal will propose recommendations for activities that will result in advancing therapies for rare diseases to reach BLA and placing other therapies in an accelerated path towards BLA. For the 4th goal, on the other hand, we seek to propel therapies targeting diseases that significantly affect Californians to later stage trials. And the objective is to accelerate the timeline to later stage clinical development for therapies that target diseases affecting Californians. So we've done a lot of work to get to those recommendations. So we are going to delve now into the data and the process that we follow to make recommendations to achieve these goals. So we will go over the high-level questions that we considered for each one of these goals. So those questions were the questions that a large team hammered down and with feedback from members of the Science Subcommittee and the Neuro Task Force over the past months, and that with answering those questions with data, we could then make some recommendations. And then we'll present a data and analysis, and then the recommendations, and then we will have a discussion. So as a reminder, the goal is to advance at least X rare disease projects to BLA. And the high-level questions that we went through were with regards to the current portfolio. The purpose here of these questions was to assess the alignment of our existing projects with our rare disease objectives and to forecast the potential impact on CIRM's funding. The second type of questions were about infrastructure utilization. Here was the focus to maximize the utility of our current infrastructure while identifying any new investments needed to overcome unique challenges in therapy development, in rare therapy development in this case. In terms of approach, here we explore the mechanisms, what are the potential strategies and methodologies that can drive the successful development and delivery of these rare disease therapies. And lastly, we thought about partnerships, what kind of collaborations, if any, do we need to identify and engage with in order to accelerate the development and approval process for these rare therapies. For goal 4, which is to propel X therapies targeting diseases affecting Californians to later stage trials. The type of questions, again, were about disease impact, what diseases are relevant to the California populations are amenable to cell and gene therapies. What are the challenges? If not, are there any challenges that we can overcome based on cell and gene therapy research to move this research forward? In terms of the current portfolio, same as before, we need to assess the current state of our portfolio with regards to these potential diseases and particularly in terms of its relevance to California health concerns, and identify barriers that might be impeding the progress and opportunities, and then map these to the recommendations. In terms of approach, the question was to refine our approach to fund and support this type of projects, and then partnerships, the same thing. Do we need to explore potential need for partnerships and what types might be most beneficial in helping us move therapies through later stage trial pipeline? We presented this slide back when we presented the July 11th goals 1 and 2, but I will go also in detail here because for the benefit of people that might not have heard this presentation before. So our analysis and recommendations have been guided by a robust, comprehensive data set, and our approach has been both comprehensive and meticulous, ensuring that every strategic consideration is backed by solid data and real-world insights. This page shows main sources of data that we have consulted internally and externally. So to start with, we have the California Department of Public Health reports, the CDC and the Cancer Registry reports. The data collection here included several years leading up to 2023 and early 2024, covering both pre-pandemic and pandemic periods, and which allowed us for a comprehensive analysis that accounts for potential aberrations caused by unusual events such as COVID-19. Taking this multi-year approach has helped understand broader trends in health data in California, allowing us to align our strategies with the current healthcare landscape in California. Then we got into our internal portfolio data analysis. And of course, for the earlier part, the goals 1 and 2, we were more focused on the discovery and early translational. Today, we're going to see more everything, but focus on translational, middle, late, and clinical. By examining our historical data, we've gained insights into the outcomes and effectiveness of past projects, and that's invaluable for our project, future project selection and distribution, and funding distribution. So the next one was the independent research by project leads and science officers. This has been invaluable. A very dedicated team of project leads and science officers undertook a deep dive into the different aspects of our portfolio and landscape analysis, captured through databases as well as in peer-reviewed papers and research articles. The focus, besides portfolio analysis, was on cell and gene therapy amenability, biomarker needs, stem cell model readiness and needs, as well as technology and bottleneck gaps. During the July meeting, we presented and covered goals 1 and 2, and our focus was a lot about the stem cell model readiness and the biomarker needs, technology, and bottlenecks gaps. During today's call, this is taken into account, but the focus has been more about cell and gene therapy amenability, the industry landscape, and we will discuss this today. The next set of data was the global database, which has provided us with a broader industry perspective. Such information is critical to ensure that our strategy is not only responsive to current needs, but also anticipatory of future scientific and market shifts as well. Then we have the IQVIA California disease landscape analysis of diseases affecting the California patient population, utilizing anonymized patient claims data from over 1.5 billion patient interactions over the past year, matched to ICD-10 medical codes. This analysis has brought us a deep understanding of disease prevalence and management trends across California. And the insights from, this was complemented from insights from subject matter experts and health economics data to further refine our understanding of where strategic investments can be most impactful. We also gathered NIH funding and industry landscape data. And finally, we had the Neuro Task Force survey of 670 neuroscientists across the board, which led to preliminary recommendations that have been included in the overall goals and recommendations that we are presenting today. Together, these data sources create a comprehensive picture that has been guiding us and is guiding us through our strategic allocation framework. But an important point to highlight here is that the data that we show at every meeting is just a snapshot representative of all the data gathered through these data sources, because it's not possible to show all the data in 2.5-hour meetings. And that's why we have the discussion as well. Sorry. A very important slide is to show who has been behind the data analysis and the presentation. During the initial strategic allocation framework presentation that we provided an overview of how the LT, the leadership team at CIRM, had been developing the goals and the questions and data needed in order to make the recommendations. But there is another essential group of people who have been working very hard over the past 3.5 months, gathering and analyzing a lot of the data. And these people are listed here. There's our dedicated team of project leads and science officers that have undertaken a very deep dive into the different aspects of our portfolio and landscape analysis that has been captured through the databases that I mentioned, as well as peer-reviewed papers and articles, because some of the data cannot be extracted through a database. You have to go into deep dive of literature reviews. I would like to acknowledge three special people here, Dr. Sarah Taylor, Thomas Trin. Without the coordination of team members and the analysis and putting together this presentation, this would not have been possible without them. And also Dr. Shyam Patel, who besides being a member of the leadership team, he led and coordinated the global data and IQVIA external analysis efforts, and coordinating science officer analysis, amongst many other things. So all of these contributions were made in conjunction with their regular duties, underscoring the dedication of our team, and we are profoundly grateful for their commitment and excellence. So now we are going to go into the data. And before we move into that, we are going to have a quick refresh recap of our research and development pipeline of programs, which encompass the full spectrum of research and development from the earlier stages of discovery through to clinical development. So the first part is for the earlier part of our discovery programs. And what I want to mention is R&D Discovery 0, these 4, these 5 is not included here. That's foundational research. These 2 is actually considered early translation. So our early translational program through these 2 offers an early translational program that supports activities that will lead to the selection of a novel therapeutic candidate or biomarker. This is new. That's ready for translation. And the expected outcome of these 2 awards is to produce a project deliverable that can immediately progress to translational activities. As a next step in our pipeline, CIRM offers currently 3 translational type of opportunities. that they focus on preclinical stage activities to position either through TRAN1 could be a therapeutic candidate for the initiation of IND-enabling preclinical studies for IND filing with the FDA, in the development of a stem cell, genetic research, or combination, or biological or small molecule. Then TRAN2 focus on diagnostics, middle translational exercise, and then TRAN4 and novel tools for broad use, addressing critical bottlenecks to the discovery or development of a stem cell and gene therapies. So that's TRAN right now in a nutshell. Now we have the clinical portfolio. So the clinical portfolio currently includes 3 programs. CLEAN1 is actually conducting, supports projects conducting later stage translational research corresponding to IND-enabling studies. CLEAN2 awards for clinical trials, post-IND filing, and FDA protocol clearance, either at phase 1 or 2, depending on where they come in. Receive different amounts of funding, but that's already clinical trials, phase 1 and 2. And then CLEAN4, which is our newest program for awards to support active CLEAN2 projects aiming for marketing approval readiness and pre-commercial activities. So through these 3 pillars, CIRM funds the entire spectrum of research from earlier translation through to clinical development. Now, based on that knowledge, how much of our historical portfolio is rare versus ultra-rare diseases? And this information is data analysis, goals 3 and 4, corresponding for goals 3 and 4. So the data on CIRM's historical portfolio, and we will have, the next slide is going to be about active portfolio, but right now is historical, reveals a notable trend at the discovery and translational stages. There is a slight majority focus on prevalent diseases, with the distribution being approximately 55% prevalent versus 45% rare and ultra-rare. As we move into the later stages of clinical development, particularly the IND-enabling CLEAN1, and post-IND CLEAN2 activities, this trend shifts, and the focus tilts towards rare and ultra-rare diseases, with the distribution reversing to approximately 45% prevalent versus 55% rare and ultra-rare. These shifts underscores CIRM's strategic emphasis on advancing therapies for rare and ultra-rare diseases as they progress closer to clinical application and potential market approval. Now, for the current active portfolio, the trends mentioned are maintained in our active portfolio. Is about the same trend, larger proportion of the prevalent are for this CLEAN, whereas there is a shift with 54% of CLEAN funding corresponding to rare and ultra-rare diseases. This slide presents a detailed overview of the landscape of cell and gene therapy across a spectrum of diseases, and the focus on those that are significantly impacting the California population. To give an overview of diseases affecting Californians, we focused on 16 diseases. So there are 2 slides. The second slide shows the cancers that affect most Californians. We focused on these 16 diseases that are known to affect most Californians. And what we observe is that while there is a significant activity in a cell and gene therapy pipeline, as indicated in the column by the global CGT pipeline, with many candidates across a range of diseases, the majority of these are still in preclinical and or early clinical stages. As we all know, CIRM has yet to fund any project that has successfully led to an approved therapy, although we have, as I mentioned earlier on, a pipeline of projects or programs that are leading in the next 2 to 4 years to BLA approval. And we have 100 and Abla will have to tell me how 14, I think it is, or, clinical trials. But this reflects the broader reality of the field. Most cell and gene therapy efforts are still in the early stages and have not yet reached commercialization. There are a few exceptions, with approvals primarily seen in, as you can see, the type 1 diabetes, and as you will see in the next slide, for two cancers, melanoma and prostate cancer, have a cell gene therapy or gene therapy approval in the US market. This underscores the ongoing challenges and the long development timelines associated with bringing these innovative therapies to market, and the relevance to this strategic exercise leading to recommendations to maximize the impact and expedite the development of these promising therapies in the cell and gene therapy domain. And this is the slide, is a continuation of the previous slide showing the 5 cancers that affect most Californians, with the patient count and the economic burden, as well as the global CGT pipeline, compare as a reference to stroke, which, as you can see, compared to before, stroke was right here. And it's just to give a reference of how many patients are affected by the most, the cancers that affect most Californians. And again, this underscores the ongoing challenges in the long development timelines and associated with bringing these therapies to approval. So as a follow-up from this analysis, our team engaged in an in-depth analysis of the field and our portfolio to figure out what are the main challenges that are precluding these therapies from moving through the pipeline, and especially focusing on our portfolio, but also having into account like how this reflects the external landscape. And which opportunities can help us move the portfolio towards approvals. So the next slide is a summary of this, and what will help in thinking about how we plan the recommendations is we have mapped the recommendations to these challenges and opportunities, and I'll try to go step by step there. So our first opportunity is the framework established by Prop 14, which mandates support for therapies that benefit Californians. Then we focused on challenges in the research and development pipeline, and at the level of translational gaps and opportunities, there's a significant challenge in our pipeline, which is the disconnect between the early and late translational phases. We need to bridge this gap and better align the programs to ensure that promising discoveries can move efficiently from early stages to the clinical development. Additionally, as the field matures, there are opportunities to streamline these processes, reducing time and cost to accelerate the transition to clinical trials. Another challenge is development complexities for rare diseases. Developing gene therapies is inherently costly and time-consuming, making it particularly challenging to scale these efforts across the thousands of rare diseases that exist. And this complexity highlights the need for innovative approaches to make these therapies more accessible to a broader range of rare conditions. The regulatory and late-stage challenges are reflected here. As we move into clinical phases, regulatory innovation presents an opportunity to enhance the efficiency of clinical studies, and this can be achieved through master protocols that allow the simultaneous evaluation of multiple therapies for different diseases, potentially speeding the development process. Approved cell and gene therapies often faced also patient access challenges. So incentivizing stage-appropriate market access strategy development and pre-commercialization activities in the appropriate programs could increase the probability of patient access to therapies. Moreover, later stage programs often require extensive investment, particularly in CMC. These requirements can delay readiness of biological license application filings, and this underscores the need for strategic planning and resource allocation in the later stages of development. This is a new program that we have, and we've gathered feedback, and we are addressing these challenges to move this forward. So we are addressing these through both goals. All these challenges and opportunities are being addressed through the recommendations of both goals 3 and 4 that we will present in the next slides. So those are goals 3 and 4. Goal 3, as a reminder, focuses on advancing at least X rare disease projects to BLA readiness. Achieving this goal requires addressing key bottlenecks in the pipeline and implementing strategic initiatives to enhance the efficiency and scalability of gene therapy development for rare diseases. How? Through our recommendations. So, as I mentioned, one significant bottleneck in the advancement of rare disease therapies is the extensive investment required for later stage programs, particularly chemistry, manufacturing, and control, CMC. These challenges often prevent or delay BLA readiness, hindering the progression of promising therapies. So our recommendation to overcome these challenges is to increase and scale Clean4 funding. This funding will comprehensively address BLA readiness gaps in manufacturing, clinical and non-clinical research, and pre-commercialization activities. And by doing so, we can increase the speed and probability of success for BLA submissions, ultimately accelerating the availability of therapies to patients in need. The second challenge and recommendation in the challenge in the development of gene therapies is inherently costly and time-consuming, posing a significant challenge, especially when scaling across thousands of rare diseases. The traditional approach to develop therapies on a case-by-case basis is not sustainable, given the diversity and number of rare diseases. So our recommendation, and this is not new to the board, there's been a lot of work around this, to address this issue, we recommend implementing a pilot platform-based approach for gene therapy development. This approach will focus on life-threatening monogenic neurological disorders as a test case. And by using a platform-based model, we aim to demonstrate that this method can enable the rapid, sustainable, and scalable development of gene therapies that can be applied to multiple rare diseases. Dr. Abla Crisi has been leading this endeavor and has already been working on this and engaged FDA and other federal and industry stakeholder leaders, which gives us, as well as the workshop that was carried out late 2023. And this gives us confidence that this is not just an aspirational goal, but is something that we've been thinking a lot. And if approved, the recommendation could take form of a concept for board consideration between September and December of this year. So this is just to show the bottlenecks that we just covered. Goal 4 is focused on propelling therapies targeting diseases affecting Californians to late-stage clinical trials. And to achieve this, we need to address several bottlenecks and leverage opportunities within our preclinical and clinical development programs. The recommendations outlined here aim to streamline the development process, prioritize therapies that benefit Californians, and address challenges in regulatory innovation and patient access. So the first one is to streamline preclinical development programs. There is an opportunity. Currently, our translational pipeline, which includes 5 programs, that is 2, TRAN1, 2, 3, and CLIN1, is at times disconnected and redundant, which creates delays in advancing therapies to the clinical stage. And as the field of cell and gene therapy matures, there's an opportunity to streamline this preclinical development, also to align it with the infrastructure technology platform that we talked about in Goal 2, and our manufacturing as well. So to address this, we recommend consolidating these two TRAN 1, 2, and 4, and CLEAN 1 to accelerate preclinical development, incentivizing multidisciplinary collaborations and fostering rapid progression to IND. We don't mean that it will be just one program, but we are meaning to combine and make it more or streamlined. We would increase the potential outcome here would be that we would increase the probability of propelling therapies more efficiently into later stage clinical trials. The second recommendation is to prioritize innovative therapies for Californians, and this has to do with the priority and opportunity from Proposition 14 mandate that CIRM supports therapies for diseases affecting Californians. We think that it is essential that our funding and prioritization processes reflect this expectation. And we propose incorporating a prioritization mechanism within our translational and clinical programs that emphasizes innovative therapies targeting diseases with clinical impact on, with significant impact in to Californians. The next set of recommendations have to do with an update, correspond to updates in the CLEAN 2 program. There is an opportunity from the regulatory field. Regulatory innovation offers a pathway to enable clinical studies of multiple therapies across multiple diseases through master protocols. And this can greatly enhance the efficiency of clinical trials. And we recommend updating the CLEAN 2 program to support emerging novel clinical trial designs, which could include adoption of master protocols that could allow for simultaneous evaluation of multiple therapies. The potential outcome here would be that by embracing innovative trial designs, we increase the probability of propelling therapies to later stage clinical trials and improving overall efficiency of the development pipeline. The second recommendation is to enhance patient access through market strategy and pre-commercialization. Even when therapies are approved, they often face significant challenges in terms of patient access, particularly due to gaps in market access strategies and pre-commercialization planning. The solution that we propose here is to mitigate this, that the CLEAN 2 program should incentivize stage-appropriate market access strategy development and pre-commercialization activities to ensure that as therapies advance, they are also prepared for successful market entry and patient access. And this slide is a summary of what we just proposed by streamlining preclinical programs, prioritizing therapies for Californians, embracing innovative clinical trial designs, and enhancing market access strategies. We can overcome some of the existing bottlenecks and seize opportunities to propel promising therapies to later stage trials and ultimately to patients who need them. And the next slide is a summary. We are getting there, 40 minutes presentation. The proposed changes to programs. On the left, you have the current programs as they are, and on the right, how the recommendations for goals 3 and 4 would affect or would change the current programs. So CLEAN 2 and 4 programs, we are currently prevalent rare and ultra-rare diseases are eligible for the same funding opportunities. And we could be prioritizing innovative therapies for diseases that affect Californians. We could also pilot a rare disease platform program with a focus on rare and ultra-rare diseases and requirement for academic industry leadership partnerships. The CLEAN 2 supports individual clinical trials for single candidates and supports a subset of pre-commercialization activities. And with the recommendations, we could suggest to support innovative clinical trial design and incentivizing market access strategy development and pre-commercialization activities. For CLEAN 4, the funding is insufficient for all activities needed to reach BLA readiness. This comes from feedback and experience with the first set of awardees. So Clean for funding, we recommend to increase and scale to comprehensively address BLA readiness gaps. In terms of the multi-program preclinical path, we have currently separated these two Trend 1, 2, 4 Clean 1 programs with their own applications that, as we mentioned, have a disconnect and are at times redundant. And prevalent rare and ultra-rare diseases are eligible for the same funding opportunities. We could propose a consolidated preclinical program, and the details of that could come through a concept or two, and prioritize innovative therapies for diseases that affect Californians again. We're going to go through the discussion and next steps now. Let me see if I have, so I can go, well, we can go through the next steps at the end, Mark, if you want. The next step is basically talking about these and these, but perhaps we go through the discussion now. What would you like to do?
Let's go through the discussion, and your timelines are extremely relevant to the fact that this is an ongoing process, and JT is likely to have additional comments around this, but just as from a super top level, not only are we attempting to ensure we're in full alignment with the mission of CIRM, which has to do with transformational work, including work not done by other organizations, as well as the ability to have a broad scale impact along with creation of leverage. So elements around protocols that would indicate multiple disease opportunities, streamlining our own internal processes for approval, resolving gaps in continuity. All these things have a direct exposure to that. And within the overall process context, not only has there been a massive amount of work done, but this is a situation where we need your direct involvement here and direct feedback, because of the fact that it's so essential that we do the best job we can in order to prepare something for consideration by the board, which is also a process for assessment and evaluation. So we need your collective wisdom to dive in on this. And now is the time to comment and review. You're going to need likely some time to digest this a little bit. So follow-on comment and review is more than accepted. But the idea then is to then have a further dialogue as we go forward into September, with the board. And again, those are dialogues. That is not, hey, the staff is presenting a plan in concrete that is not to be gainsayed in terms of discussion. So I think that goes without saying, but I just wanted to articulate that, just for avoidance of doubt. So JT, I don't know if you want to amplify that or any other comments you might have.
No, I think that's very well said. This is as meant all along to be a team effort between the members of the internal team and the board, and so nothing whatsoever is cast in stone. These are, I say, merely a set of goals and recommendations. That's merely would suggest not a lot went into it, which of course is certainly couldn't be further from the truth. But this is something that we want everybody's input on here and at the board, so that we're as informed as we can be when we get to the decision-making process that will happen at that meeting. So I'll leave it at that, Mark. Thank you.
Great. And just one other comment. I'm beyond impressed with not just the massive hours that have gone into this, but also the hyper quality of the work. In fact, I'm convinced some of this could be written up for other organizations to follow going in the future. That's how seminal I think that what the team has effectively accomplished. But I'm also impressed by the broad scale teamwork within the organization, and we're all part of that team. So it's extremely important that we provide our feedback both today and in the coming weeks as we go forward. And with that, I'd like to, Carolyn, if you have some comments.
Yeah, thanks so much, Mark. I also wanted to echo that this was an incredible amount of work and careful analysis by Rosa and the team. I'm wondering if it's so much that was presented. I'm wondering if it might be helpful to go to the high-level questions that then the data supported to have the board have a little bit of input into questions around rare disease, prevalent diseases, how we would address some of the more challenging hurdles, whether it would be consortium grants, the partnerships with industry, all of the rich ideas that were presented, but maybe get some feedback on those. Just suggestion on how we might proceed.
Okay. Should we just jump into that? Would that be the right approach?
I think Monica also had a comment.
Monica, you've got some comments here.
One, just have to echo, this is such an impressive thing. And I have to say the comment that this whole process should be published should actually be taken quite seriously. This is such a big thing and a process. So I really would just like to double down on that. I am sure this has been considered, and in the magnitude of data that you have presented, you can't present everything, but just sort of to get it on the record kind of thing that you have thought about these things. When we talk about all Californians, California, we're not a homogeneous state. We're a highly heterogeneous state regionally, socioeconomic demographics, various racial ethnicities, groups, and all of that. And the health disparities and equities between those are great. And then lastly, when we're considering things from my field, when we're looking at various GWAS databases, patient claims, they are not evenly distributed around the real incidence of diseases and how they're recorded. You can't do all of anthropology, sociology, and medical anthropology, but I know that you all have been very considered. This may not be the time to put it up, but I think that should also be part of the record and the considerations of how these things are prioritized and also understanding the challenges of making impact on some of these things as well. But thank you.
And then before we launch into the some of the follow-up elements, Maria, do you have a comment?
I know I just wanted to, it has been an amazing amount of work that the team has undergone. And for that, I am just very thankful and just very proud of all the work they've done. So it's a huge challenge to get all this data together. Kudos to you, Sham, and to be able to present this in a manner that that even me as a non-scientist can understand and relate to. So thank you. And I wanted to echo Monica's comment. I think arriving at how we prioritize disease relevance and how we come about getting to what the number X means, or the letter X means, what number that relates to, I think is going to be a really big challenge. And so those are just some questions that I would have moving forward.
Yeah, no, and I appreciate this. I think, thank you, Maria. The definition of the X is still being refined in collaboration with feedback from board members, and we would appreciate like if you want to send us emails or connect with us to discuss this. This will evolve as the strategic allocation framework prioritization process continues, and we expect that we will have the concrete information to our September meeting, the first one, the Science Subcommittee, Neuro Task Force. We would have some numbers. I think it's easier to add numbers to this incognito, this X, for the clinical actually. It's harder for the earlier stages in terms of technologies, or that we will validate, or technologies that, or identifying new targets or biomarkers. As we all know, that's very hard, and especially validating them. So we want to be very mindful the way we do it. And I don't know if I got the question right. And Sham, feel free. Sham was the holder of the baton for all the external data, but in terms of the diseases, while diseases with large patient populations and significant impact are certainly important, CIRM will also prioritize unmet medical needs and innovative approaches that can have a transformative impact. And what we are talking here was more at the level of the how we were thinking in terms of the recommendations, but each project, when it's evaluated in its own merits, including the potential for broad applicability and significant patient impact. We just wanted to have an idea through all the databases that you've seen that we are not suggesting a list of priority diseases. We just wanted to show, CIRM is derived from Prop 71 and Prop 14, which is for diseases that affect Californians. We wanted to have an idea. What are the diseases? What's the amenability? Where are the gaps? What can we do to move things forward to have an effect on Californians?
Okay, before we get back to Carolyn's question, let's go. I see Monica and then Fred Fisher.
I just want to do one quick clarification on one thing. That was really beautiful. Thank you for clarifying that. I would suggest the clinical data is not as easy as said. Our databases are structurally biased, and the algorithms for medical treatment, the way we record people's complaints and we bin them. When I started in the field, folks from Latinx, Latino groups don't get multiple sclerosis, and it's because of how we recorded things and the presumptions that they're in. So this is what I'm saying in my fields, and I've part of another group in my professional life. These databases are difficult, so I don't think it's as easy, and I think that's what I just want on the record. But we're not going to solve it here. It's just an awareness issue.
Great.
Thank
Fred?
Yeah, if I, again, want to add my gratitude to the team for putting this report together. If I understand correctly, the goal is to shift, at least in some way, toward later stage research funding. And if that's true, the later you go, the more expensive it gets. And so the pace at which we are consuming funds would likely increase as we shift our priorities toward late stage support of potential drug candidates and working with drug companies. And we just may want to consider the financial impact of moving to supporting late stage development, which has been primarily supported by these commercial companies going out and getting money. And if we are going to become a player in that space, we're going to be spending a lot of money, a lot more than we have on a project by project basis. So I don't know what to do about that, but just want to put it out there.
I think it's a very relevant point, first of all. Second, I think within the discussion points here, one of the key framework elements has to do with the fact of involving a lot of different groups, first of all, and second, including dragging in large pharma at an earlier stage in the process, with a concept being that we could throw a ton of money at stuff and it's going to be a drop in the bucket for actually ultimately achieving commercialization of some therapeutic, let's say. And so within that purview, then can CIRM be the spark plug to pull this all together and to make it actually happen that otherwise would not have happened on its own. But clearly, Fred, you're spot on. And whatever allocation mechanisms are determined here has to be done in the review of, well, what are the dollars allocated to that, and is that our real intent related to that? So your question is front and center as part of this process overall from my perspective.
Thank you.
So can I just clarify, if you don't mind? I think this is a very good comment, but I just want to make 2 points clear. One is our goal is not to move towards the later stages. We are not de-emphasizing basic research. We are streamlining things and focusing. So streamlining means for discovery, as we showed, we could be going through the multidisciplinary, we could focus on bottlenecks through technology platforms, but we would select in collaboration with industry the most relevant bottlenecks and the things that we have resources to leverage in California. And we would align this with our development pipeline. For what we presented today, the goal is not to move towards the later stages and invest more. We have received, and I was mistaken, not Clean 4, but from the Clean 2 applicants, we've received feedback that there is a need for Clean 2 awardees to in order to support BLA readiness. The costs are higher, and like the activities that we would need to fund would involve some aspects like later stage product development, pre-commercial manufacturing, as well as clinical and non-clinical research potentially. So we are enabling, as we have a pipeline of potential programs that are going to BLA readiness approval, we are allowing for those potential applicants to be successful, and it could be a stage type of a scenario, but that could be for things that we could prioritize. And Sham has his hand raised, and he's been developing this, and I also would welcome Abla's comments. Please go ahead, Sham.
Thanks, Rosa. I just wanted to reiterate that point, and as well as the overall goals. So with respect to the later stage development, this is for the CLIN 4, where existing CLIN 2 awards have some gaps they need to overcome in order to get to BLA readiness. And as we know from cell and gene therapy development, more broadly, those can be substantial. And so this is addressing a need in our own portfolio where there might not be other resources that some of these programs, particularly those in the rare disease space, can tap into outside of CIRM. So that's really the intent, is to support those programs more fully for the CLIN 4. The other goal, just want to clarify, so we've been thinking about this as the impact of CIRM with the proposition money up until the last award is executed. And so in that sense, if we're going to be focusing on that, can we do things along the spectrum of therapeutic development that could result in higher efficiency of candidates going to clinical development, and then demonstrating some sort of clinical proof of concept that can then progress them into late stage development. So we're addressing the things along the way to get better candidates into later stage clinical development, but not necessarily suggesting that we would prioritize only late stage clinical development in that space.
Thank you, Sham. Very well said. Yeah.
So if I could just quickly
go
respond
ahead,
to
Fred.
that. Thank you. So I'm trying to put this in the vernacular that I'm more familiar with, which is looking at the drug development pipeline and figuring out where we're deploying our funds. And it seems like we're talking about kind of valley of death funding to get a project over to the point where they may be able to attract more commercial money. And that's an expensive proposition. It's why a lot of companies fail, because they cannot attract the money. But it's a significant amount of money. And I would love to see a graphic that's different than the one that you put up, which is sort of CIRM related. I would love to see a graphic that sort of deployed these initiatives and where they are targeted in the drug development pipeline. Because that would help me contextualize what we're really talking about in terms of where our money's going to actually go. So if that's possible for some future meeting, or even before, I'd appreciate that.
Yeah, I'm not sure if absolutely. What I was wondering is if this, let me just go back to the slide that shows the within the 1 second, Fred. If it's something else, I just want to make sure that we have it ready for the next meeting. So in this specific graphic, it basically portrays that in these two TRAN 1, 2, 3, 4 clean one, this is one of the changes. We would combine these and probably have just two programs. Also, it could have a prioritization for diseases affecting Californians, and there could be where the rubber hits the road is going to be in the specific criteria for review and eligibility. So that's going to come when we bring the concept. And I think what we are doing, yes, so that's number one. So this is one of the recommendations. The other recommendation that we brought in was to increase Clean 2 funding to provide the novel, what was it? What's the name?
Platform?
It's the platform format trials to be able to assess and the master protocols for multiple therapies. So that's number one. Right now we do single therapies, and then there is the funding for the rare disease pilot platform, which is also it could be around here as well within Clean 2, and then we ask that the Clean 4 funding to provide for more funding. However, this is going to be only accessible to successful Clean 2s that are ready for BLA ready activities. And of those, the board knows from the closed session how many we have for that. We have two hands open, and I would like to lead to my colleagues, Sham and Avla.
You want me to go first, Avla?
It's okay, go ahead. You were ahead of me.
Thank you. And so just want to support Rosa's comments and point out that the traditional, the development pathway that Fred, you rightly asked about is shown here as well. It's a little bit hidden, but so if you look down below the single product candidate, pre-IND meeting, IND approval. So essentially what we're showing here is that the discovery stage activities, the preclinical valley of death activities, IND filing, and that initial clinical trial, those are all the activities up to the end of phase 2 meeting or equivalent, right? So those are all the traditional valley of death activities where you have preclinical development and early clinical development. And most of our recommendations are in that range of activities, because that's predominantly where the CIRM portfolio is also focused. So we're here proposing to make some changes that will help create more efficiencies, as well as take advantage of multiple approaches and multidisciplinary approaches for the development along that paradigm. So basically just making it more efficient in the way that we fund these programs at the stage we're currently funding them at, which is that preclinical and early clinical development.
Just to echo what Rosa and Sham said, in fact, we already have data where some of our grantees are not needing as long as the amount of time in order to stay in a DIS2 or a TRAN 1 to get to the pre-IND meeting. They're jumping fast towards filing their IND and moving into a potential clinical trial. So instead of kind of boxing them into, you got to be in Tran 1 for X number of months, for so many years for Clin 1, et cetera, it'll be important to recognize that the trend is that they're not needing as much time and more data in order to get to a clinical trial. So given that information, that allows us to become more efficient, not necessarily increase the amount of funding. There may be, we're still the ones who are already in the pipeline in a Clin 1 or a Tran 1 can continue, but we're providing the option of having that flow from DISC-2 to filing an IND and getting a clinical trial going can be allowed in order, since we're seeing it happening in front of us, and that's from a practical viewpoint. We recognize the importance also of needing to change or at least stay abreast of what the regulators are doing relative to clinical trial design. And in some ways, it will help our grantees get closer to identifying the right candidate to move forward using a Clin2 that has these master protocols, whether it is an umbrella trial, basket trial, or platform trial. So the key here is not really to expend our dollars, but how to stay current with the regulatory innovation as well as with the needs of our grantees. And again, I think we need to stay close to sort of like adapting to what is happening in our industry. And we recognize the fact that we don't really want to spend our money, without understanding the trends that we're seeing. So thank you for your comment, Fred.
Hey, I'd like to do is turn the Q&A over to Rosa. I'd like to get back to Monica's question, which had to do with some contextual framing. Monica, if you want to, no, sorry, Carolyn's question, my mistake. Carolyn's initial question related to, some context around definitional aspects. So Rosa, I think your initial slide, but I'll turn the Q&A over to you, so I don't need to moderate that.
You turning it to me? Oh, I thought you turning it to Caroline. Yes. From my understanding, Caroline, your intention was to go over these questions and make sure that we have covered that all these questions are sufficient, or if there is anything else that the board is thinking that we should be thinking about answering. Sorry, it's moving with my, there you go.
Yes, either the high level questions or the approach. You've conveyed the data very, in great depth. Just wanted to see if there was further input. It sounds like we've already had a rich conversation, but it was just a suggestion.
Yeah.
Mm-hmm.
I could go through, our rationale. We had many more questions. I can also go through the process, how we did this. I don't know if it would be helpful, but about the whole LT gathered for several days through a period of a month to go over first defining the categories and then defining the impact goals, and those were initial impact goals that we defined, but we had the 4 categories very well established through the strategic plan that we already have. And then we went into thinking and brainstorming about what questions and what sections good we have to cover in order to figure out how these recommendations could be justified if we made that. So one of them, the first one, is the current portfolio. And maybe here we didn't say it, but it was implicit that not only the current portfolio, but we also did landscape analysis of the industry, on the portfolio, the general portfolio on rare diseases, and the regulatory landscape as well. You know, how many these cell and gene therapies have been approved, which diseases, how has it changed? All those things were taken into account. These are very high level, representing what we've shown today, but there was a lot more granularity. So in terms of if we want to advance at least X rare diseases projects to be like, what do we have in our portfolio, right? And also we want to know how effective is our pipeline. How is our pipeline going from discovery to clean 2? What's the proportion of projects that go from these 2 to TRAN, from TRAN to clean 1 to clean 2, and how does this compare to the industry standards, right? Are there any challenges there? So that's the kind of things that we evaluated. In terms of infrastructure utilization, we looked at, you know, if we want to do that, is there anything in manufacturing? Manufacturing, we've only had phase 1. And Sham has been leading these, and he's now, you know, he's going to plan for the next phase, right, Sham? But not only this, but also the patient support infrastructure, and this links to the next goal 5 that we will come with in September. So things like that, just to show that it's all connected, right? And then with clinical, that is what derived to some of the outcomes that Abla was mentioning in terms of increasing the funding for some activities that are not currently funded through Clean 2 or through Clean 4, right? In terms of approach, you know, how would we have to do this? And one of the things is in part like more operational, or how we have things. That right now with the discovery, with the preclinical research, we realized that historically we've had these 2, the TRANS, the Clean 1, but is a time for reevaluation. Also, we want to look at the current portfolio and maybe have an audit of the current portfolio, right? I don't want to scare anybody, but we do need to look at what it's there. There are clinical programs that maybe we need to look at them and say, well, you've been doing this for a very long time. Are you tackling the right bottlenecks here? Are you really like going to get there? And if not, we might have money to invest in the kind of things that we are trying to invest to move the pipeline forward. So those are the kind of things that we are going to be, yes.
So, and I wasn't questioning how all of the data was put together to answer these questions, just to see if there was further feedback on the presentation and on maybe some of these questions around partnerships, around the data center, all of that. But maybe it's not that helpful.
Yeah, you were opening for other members of the board, not to provide their input, Caroline?
Yeah, just to see if there was more input.
Yeah, no, and I was actually
presentation.
Stop sharing. I was actually just mentioning so that people could have time to think about what they might want to say as well. So I was going through this so that they had a bit more time. And maybe by me explaining our process, it could lead to somebody thinking, oh, this is how we connect. And one of my colleagues, Abla, was mentioning that you know, we want to accentuate, and JT also mentioned this, that the discovery part of our pipeline is, as we mentioned in goal 1 and 2, is extremely important, right, as well, because we are not a venture capital firm here. What we are is a funding agency, and we need to figure out how to strengthen our research to lead to overcoming bottlenecks. Some of the things that we will leave as a legacy will not be an approval, but will be a biomarker that will help us accelerate a clinical trial in the future. You know, some of the bottlenecks for some of these neurodegenerative diseases, which might not be amenable to cell and gene therapies, are that they are very long clinical trials and nobody can invest in them, or they're very expensive clinical trials because we don't have the right biomarkers or the right biomarkers to stratify, or we are not going through the right system or target, right, or disease mechanism. So those are the kind of things that CIRM is also should be focusing. And one of the things that, we've heard it lately, we need to message it properly to California, right? Because this is the value that CIRM will have in the future as well. And we have to be able to show that early research, translational research might not be at the end of CIRM Proposition 13, leading to an approval, those specific, but we need to invest because they will help in the future. So I don't know if there is input in these questions, as Caroline was suggesting. So I guess we did a fantastic job now with the questions. Fred is going to give us some input. Fred.
Some more to think about. So when I look at this, I'm wondering, like the very first question, what proportion of current portfolio supports rare diseases? You showed us a very comprehensive slide that breaks all that down, but it forces us to answer the question for ourselves. And as board members who are not as familiar with this data and don't have the time to add up all the bar charts, it would be good if we just had an answer. And there's other questions that just sort of lend themselves to just, if we have the answer, let's provide it. Because we don't want to, people are going to see this for the first time or second time or third time, but they're still not going to be able to manage the data in their head.
Yeah, so that's a great point, Brett, and I appreciate, because we have been looking at this so much that we don't realize that it's not. And in my narrative, I mentioned what it was, but I think this is a great idea. We should have a little pop-up here that says, in the earlier stages, this can run. Prevalent diseases are more prevalent in our portfolio. So we have a 55 to 45% ratio. So 45% in rare and ultra rare disease research and 55% in prevalent. And that's mostly because of what we showed in data and goals 1 and 2. There are models, stem cell models for many diseases that are prevalent that we can utilize to model the disease and do more discovery and validation of findings in the early stages. Now, those diseases are not right now amenable for cell and gene therapies. Cell and gene therapies are often designed to address underlying genetic or cellular causes of diseases, which are particularly well suited for rare and ultra rare conditions, where specific mutation or cellular dysfunctions are identified. So as we advance, we will be able to tackle more prevalent diseases. So this is what this is showing, right? So we will add a little pop-up that says the proportions, so it's easier, and we will do it in the next one as well. Maria, your hand is up.
I wanted to ask questions around partnerships. If you could go back to the slide.
Yes.
Had partnerships as one of the our strategic partnerships necessary to achieve this goal. I mean, I think that's rhetorical, but I would say yes, absolutely. And it's, we've done a good job in the past of sort of identifying areas where we could partner. I think we could do an even better job moving forward. And I think that it's, a skill set of someone being able to connect the dots of, constantly combing through information of who's doing what and then relating it back to either initiatives or potential initiatives that we ourselves are undergoing. So I think that's key moving forward. I think partnerships are absolutely key. One allows other people to know who we are and what we're doing. So from a PR perspective, super valuable, but then also from a money perspective, also super valuable, because somebody else has skin in the game and also extends our dollars longer so that we can engage in more research and activities that help move this along the continuum. So I'm a huge proponent of looking for more of those partnerships.
Yep. Thank you, Maria. That's a great point. And JT.
Yes, I want to, on focusing on goal 3 there, as everybody knows, received a letter, very well considered letter from a great many from the disease advocate and patient area that was concerned about rare disease and how we would be focusing on that going forward. And I think, Abla, if you could give a little extra color on that for those members of those very important groups, to let them know the different ways that we will be considering rare disease. I think that'd be very helpful. Thank you.
Okay, thank you, JT. Yes, again, as a rare disease patient advocate myself, I just want to emphasize that we will have 3 different pathways to advance rare diseases. One is the current portfolio has a number of rare disease grants that are advancing towards late development and potential BLAs, and we're going to support them wholeheartedly with that kind of CLIN4 type of grant where we're advocating for enhancing the funding for that purpose. We found out that it's probably the burden of filing the BLA with current grantees is around 25 million. We're supporting a CLIN4 with 12. And so for that reason, we're saying, okay, we heard you, rare disease, we'd like you to get approved. And for that purpose, we're going to see if we can enhance the chance for you to do that without having to come to CIRM twice to do a CLIN2. And that's really the rationale for advocating for getting CLIN4 funding increased. The other is that, as you know, when it comes to clinical grants, we started the granting process again for CLIN1s and CLIN2s at the end of July. And the entry to an application to CIRM through that route is wholeheartedly open for rare diseases as well. And so please be mindful of that. And when you're ready, go ahead and apply. Despite the fact that this time the number of months before anyone knows whether they got awarded the grant or not is now prolonged by 1 month, but it's still worth it. And we're ready to evaluate whatever disease you're advocating for and what kind of grants are you proposing. The third path, which is the path that was discussed a little bit, is about potentially developing well, it's not potentially, we're actually have progressed our thinking there. We're going to develop a rare disease pilot as a consortium where we're going to enable the development of rare diseases in an accessible, scalable, and sustainable manner in collaboration with multiple groups outside CIRM, as well as with the regulators. And that exercise is going to allow us to be able to integrate a number of really good things that the regulators have already identified when it comes to regulatory innovation per their variety of guidance documents. For example, if it is a rare disease that is serious and life-threatening, the preclinical package doesn't have to be lengthy. So how can we use that in the design of the platform? I'm just giving you examples. The other would be, once we have, as many of you know who are working in rare diseases, we recognize that whether it's the biologics branch of the FDA or the drugs branch, they're both are advocating for doing a rare disease natural history study or a registry, and you no longer have to do sham or placebo-controlled trials. So, and we're mindful of that as well. I just want to give you examples of why we think this rare disease pilot platform type technology an approach is really going to build on a lot of advantages that the regulators have given us to allow us to move forward in a more expeditious manner and solve the issue with many of the rare diseases, especially if we were to start with the neurological diseases, with which many of you have already shared with us, that when it comes to rare disease, probably 80% of them are genetic, and many of them are in the neurological area. And so if we were to capture all what we have learned thus far in that kind of a platform technology, then it's going to allow us to address the needs of the patients much more quickly, most likely with equal dollars or less dollars per grant, in order to allow us then to create like an avenue for then doing more than one patient or two patients at a time. We can do 10 at a time, 20 at a time, 100 at a time. So that's where we're heading. We're not ready yet to put together a concept plan, but we putting that all these aggregate, whether it's technology, innovation, regulatory innovation, as well as readiness in pulling all that data together in where everyone shares their data readily with each other, is going to allow us then to be very much the real advocates for rare disease and allow us to utilize whatever dollars we have for rare disease and common diseases, utilized in a manner where whatever lessons we learn from such a pilot can be applied for the common diseases as well. So if we were to develop a roadmap that shows we were able to problem solve for like a number of those rare diseases, and we identify in the meantime a target for a common disease, can we do the same for the common disease? So just would like you to think also what we're advocating for is to utilize what we built in terms of our infrastructure. Use the alpha clinics, our manufacturing units, et cetera, to help us move the programs in a consistent manner, hand in hand with the regulators, to allow them to succeed and reach the patients. So I'm happy to answer any questions you have relative to, again, the three different paths. And once we are ready for the platform technology type consortium, I'm happy to engage all of you in the discussion with us.
And Rosa, I would just add to what Abla said that, and of course, rare disease grant applications in the discovery and translational phase, or the condensed new preclinical phase, will continue apace as well. So
correct, yeah. I just would like, I always say something along the line, think of a conveyor belt. Look how the automobile companies have started doing, putting together the car engine, and then, let's say actually that analogy was used with us. The FDA put together the car engine, but we need to put together we know how to put together the doors and the tires on some of those platforms. And so that's what we're trying to do right now, but in a very amenable way that will be practical. And again, that would allow for us to do accessible, scalable, sustainable, and affordable rare disease therapy modalities.
Thank you, Abla.
You're welcome.
So I think JT and Caroline and Mark, we were in the framework of the questions. If there was any other, I can go into goal 4 if there are any other comments, questions, additions to these framework questions to make sure, and I appreciate very much Fred's suggestions to make it more like processable as we are presenting. This is a lot of information, and as we move forward, we are going to have 6 goals at the September meeting, and that's going to be quite dense. So we want to make sure that we have the information in a way that's easier to process. So any others? Mark? No? So if there are no other questions or comments, what we can do is show, if it's okay with you, Mark, the timeline next steps and where we are and where we will be soon. Sounds good?
Sure.
Great.
And if anybody thinks about questions in the meantime, jump in. This is the time to ask.
And also we are available anytime to receive feedback on the numbers or have a discussion between now and the September joint neuroscience subcommittee and the Science Subcommittee and the Neuro Task Force. So today we went through the feedback on goal 1, 2, introduced 3 and 4 and discussed the data and the recommendations. And at the September Neuro Task Force Science Subcommittee, I believe, Claudette, it's September 13, correct? That's correct.
Yes.
Thank you, Scott. We will have the full presentation but we will make it a little shorter than the ICOC. We are going to present 1, 2, 3, 4 with the feedback received, and then focused on goals 5 and 6. 5 is access and affordability. 6 is providing opportunity for us that strongly focuses on the education recommendations and training programs. And then we will discuss the overall recommendations and any comments in preparation for the September ICOC. I also believe, Scott, do let me know if it's my task to say this or is Mark's or yours with regards to seeking an endorsement from, is that your role or Mark's?
Well, I suspect that, it would be helpful to the full board to have a recommendation, an endorsement from the joint subcommittees with regard to the goals. So I think that would be helpful at the conclusion of the process to seek that endorsement.
Thank you. And then in terms of timing, this is the meeting we are talking about, that we will be seeking an endorsement in preparation for September 26 ICOC, which is in San Diego, correct?
That's correct.
Yes.
And we will all be there in person to present these and other things, but this is a very important part of our history, I think. And with that, any other questions? I'll leave it to Mark to close the meeting.
Yeah, with that, there's obviously a lot of material here. I would really encourage you to look through it and to give it careful consideration. This is a very important time to be able to ensure that the proper questions have been asked, that the consideration of additional feedback is taken into consideration. And obviously we have another bite at the apple here in September, but I appreciate both your collective wisdom and your collective effort on behalf of ensuring that as an organization we can move forward in the best way we possibly can. So I will be very much, I'm very, very thankful and appreciative of your efforts in this overall process. So thank you very much.
And Mark, before we adjourn, we'll just want to solicit any public comment once the board is finished.
Great. Any other questions from the board before we open it up for public review and comment?
Just want to say thank you, Mark, for your leadership in moving all of this forward. It's terrific.
Well, thanks, Fred. I'm doing nothing. The team is unbelievable. So it's fun to surf the wave here. So yeah. So thank you. And with that, any other? Oh yeah, Brenda, is that? I'm sorry,
I'm not on the board. So are you ready for public comments?
Public comment. Yeah, hold on just a second. Just any last minute comments or questions from the board. And then Claudette, if you would let us know who's in the sequence for public comment.
We're starting with the two we have in the room, which are Nasha and Leah, and then we'll move on to Brenda, who's on Zoom. Everyone has 3 minutes. I will keep time. You will be able to see it internally in the room on the screen and also on Zoom. All right. Okay,
great.
thank
Claudette,
you.
I'll let you control the flow on that. So thank you.
Okay. All right. Thanks, Mark.
Should I start?
Yes.
Okay. All right. Hope you guys can hear me. Can you hear me? Yes. Okay. All right. So thank you so much. And thank you, John, Abla, and Rosa, for the words you've said about rare diseases and your work in this area and for engaging with us. We really appreciated that. My name is Nasha Fitter. I'm a California resident, taxpayer, and mother of a daughter living with an ultra-rare disease called FOXG1 syndrome. And this condition does have the potential to be treated with a gene therapy. I am here representing all California families within the ultra-rare disease community who are tirelessly working towards creating treatments. For us, support from CIRM is vital, as federal, pharma, and VC funding is almost non-existent for our conditions. That leaves the job of fundraising, unfortunately, to patient advocacy groups like the one I lead. And I will tell you, despite my extensive professional experience in fundraising and navigating capital markets, I have faced a really harsh reality that it is brutally hard to raise money for ultra-rare conditions. Just this week, a senior biotech executive told me, and I quote, "We're not investing in ultra-rare anymore. Our last program didn't make enough money. We're going to focus on much larger rare diseases." Yet as parents, we have no choice. We are not going to let our children die or have a lifetime of suffering. Our foundation united our community. We invested in basic science. We built essential resources like a patient registry, like a natural history study. We've raised funds through grassroots efforts, and we have published data on our gene therapy preclinical trials. But now we're at a pivotal point. We have an extremely promising program that's ready to enter GLP toxicology. The next stage is clinical trials, and as you all know, they come with a price tag of $10 million, an amount that is simply beyond our network's family and friends' reach. Like so many others, we face the threat of sinking into the valley of death, and not because our science has failed, but because of a lack of funding. CIRM is the best and perhaps the only avenue for funding that small biotech companies and patient advocacy groups like ours can turn to. While larger conditions continue to receive billions of dollars in pharma and VC support, ultra-rare conditions like ours are left behind. We understand that CIRM needs to maximize taxpayer dollars, and thus I just urge you to think about this in the following ways. First, should our taxpayer dollars be invested in areas ignored by capital markets? Should taxpayer dollars be used for conditions that could find funding elsewhere? Second, the majority of ultra-rare diseases are monogenic. They are easier to target and treat with gene therapies. Shouldn't taxpayer dollars go where there's a higher chance of success and
Claudette, you probably should extend her time.
And we have more of a chance to be innovative with clinical trial design, which is a huge goal for CIRM. Thank you so much for hearing us and for your partnership with our community.
Sorry. Thank you so much. Leah?
I don't have any comments.
Okay.
I'm just here to learn.
All right, so we have next Brenda.
Hello, I'm Dr. Brenda Porter, and this is my colleague, Dr. Emily Spellbrink. We're both pediatric neurologists at Stanford. I'm a professor of neurology and I study pediatric epilepsies. My clinical load is quite high and I have a large patient population, as does Dr. Spellbrink, with pediatric epilepsy. And our patient population is really lots of patients with lots of rare diseases. So we basically take care of kids with tons of different disorders that are generally pretty severe. And this was fantastic. I had never been to one of these CIRM meetings, so thank you. Really interesting work you guys are doing. One of the reasons we wanted to speak was really to focus on that issue that we have where all of our patients have rare diseases. And we have drug companies come to us with therapies, but they're not usually targeting super rare diseases, but that's the majority of the patients we have. So I think it's fantastic that you're considering continuing in that space. Two things I'd like to kind of point out was, long ago when we would get NIH funding, we all had our own IRBs, and the NIH said, nope, you're going to use a central IRB. And that was a huge help to all of us who were doing trials because it took forever for our local IRB to approve things. And so once the funding agency said, you are going to use a centralized IRB, we had much better success in, getting patients into trials quickly. So I think you are in a great position if you're funding some of these trials in rare diseases to really think through what you want our institutions to get out of the grants and what you want the patients to get out of the grants and how we can interact with companies. Because in this rare disease space, just like was mentioned, it's super hard to get companies interested in the disorders. And it's tricky for us to walk the line between Stanford and these rare disease companies to make sure that everybody is benefiting, but yet moving forward quickly. So it would be fantastic if you guys could think through that a little bit. The other thing is I hope that you'll do a combination of funding of really early, radical new ideas on how to get gene therapies into patients, but also sort of look at the current pipeline out there, such as AAV9s, which are really probably the standard right now, and fund both. Like think about it as trying to get us to the future, but also what's available now. So thank you. This was really fantastic.
Okay, Claudette, I don't think we have any other public comment.
I do.
Okay, sorry.
Can you hear me okay?
Yes, we can hear you
Anyone
now.
able to hear?
Yes, can you hear us, Justin?
Yeah, can you hear me okay?
Yes.
All right. Thank you for the opportunity to speak and for the vital work that you're doing at CIRM. My name is Justin West. I was born, raised, and practiced medicine in California. I come before you today as a rare disease advocate and the exhausted father of a 7-year-old boy with an ultra-rare epilepsy. My family hasn't just been to the breaking point. We live there every single day, dealing with the devastation that comes with watching a young life stolen, struggling to keep a child alive who tries to die every month, and the always looming fear of what happens if he outlives us. We're living in a golden age of science where we can identify the genetic cause of disease and quickly develop treatments to address them. Several rare disease groups have already done this. Simply put, the rest of us need funding. I believe a disproportionate amount of the current funding is allocated for diseases that already have adequate treatments and to diseases that are so complex that billions of dollars have resulted in no clinically meaningful impact. Consider some of the current funding. $50 million to HIV clinical trials, yet HIV is now a chronic disease with a life expectancy similar to those without the infection. $100 million to glioblastoma research, a disease that has seen no improvement in 5-year survival rates despite billions of dollars spent over 30 years. On a slide earlier today, type 2 diabetes was listed. I would also argue against funding treatments for diseases that are caused by and treatable with lifestyle choices. As a father of a child facing a 50% 10-year mortality and the brother of a 40-year-old whose life was cut short by glioblastoma, I asked you to consider this.
Reduce funding for diseases where the mechanism is unclear or treatment strategies aren't well defined. Stop funding programs to treat diseases that already have treatments. We see limited resources allocated towards ultra-rare diseases in which the mechanism is known, the pathway for gene therapy is clear, and there is real potential to develop therapies that can result in reduced suffering and mortality. The opportunities being missed are staggering. Unlike the prevalent disease community, a lack of funding options has forced families to take matters into their own hands. When your child faces catastrophic disability or a death sentence, you don't have the luxury of waiting for pharma. Instead, parents like me quit jobs, start foundations, assemble advisory boards, open patient registries, conduct natural history studies, populate biobanks, generate cell lines, and establish international research collaborations. We become the untrained CEOs of the only company that truly matters to us, where failure isn't measured in dollars, but in lives. For 7 years, we've cared for Andrew 24 hours a day, changing diapers, spoon feeding every meal, starting IVs when he refuses to drink, and meeting every other need for our functional 6-month-old day in and day out. Every time one of our other children hits a milestone, we die a little inside, knowing Andrew will likely never experience those moments. This is our reality, but it doesn't have to be. Shift the funding strategy for CRM could be life-changing for patients like Andrew. Other groups have gone from treatment concept to trial in less than 2 years. The rest of the ultra-rare community is desperate for support to have their own clinical trials that could drastically reduce physical and disability.
Thank you all so much. That is it for the hands raised for public comment. Mark?
Okay, with that, thank you for everyone's time. Very much appreciate the public comments, which are a very important part of the overall process and discussion. And I look forward for continuing the process here of marshaling all the activity to come to the best goal that we can possibly come to. So look forward to the next steps. And again, in advance, I very much appreciate everyone's efforts directed to driving solutions as we go forward. So thank you. So with that, we're adjourned, right, Scott?
That's right. We stand adjourned.